We are on to our final speaker, who is on Zoom. David, we are ready for your talk whenever you are ready to share your slides.
All right, let's do it. Hi, everyone. Wish I could be there. Wish I was there. Next year for sure.
Oh, that's great. I'll hold you up to that promise.
I've got my father visiting from Australia. He's 83, and I think that took precedent. But next year,
Fantastic.
I'll bring him with me.
That would be amazing. That would be amazing.
Okay. Well, thanks for inviting me. Let me get my presentation up and running.
Perfect.
All right.
Great.
And I'll set a timer so that we're good. Okay. All right. So yeah, it's amazing to be here. This conference, to your credit and the other organizers, it's growing to be one of the premier conferences in the world on this topic. So hats off to you. And I've really enjoyed the talks. So just because I don't have much time, I'll get straight into it. So I've been doing this for a while, started at MIT working in little yeast cells, and we discovered that, and by we I mean a lot of other people as well, the team discovered that the reaction to genomic instability, particularly at repetitive regions, leads to the reorganization of chromatin, which leads to changes in gene expression and phenotypes of aging. And what we work on in the lab is that process that we worked on in Lenny Guarenti's lab in yeast in the mid-1990s is also true for mammals, and that there's a backup copy of information to reset the epigenome. So what I'll do today is I'll share a fair bit of unpublished research. It's really a coming out moment here. So this is how we think in my lab. Similar to many others, we like to understand why we get old, and we think that the hallmarks are part of a whole system, and we think that the loss of information in particular is very important. And information comes in two types in biology mainly. It's the DNA and the epigenome control systems. And we have quite a lot of evidence, in the field increasingly so, that the epigenetic noise, as we call it, and the loss of differentiation is a major driver of the process we call aging and illness. So I'm going to talk about efforts in many organisms, including humans, to slow down aging by targeting epigenetic modifiers, which include the sirtuins, which we in Lenny's lab worked on and continue to do so. I'm going to talk about the ability to reverse aspects of aging, including epigenetic age. And I'm going to talk about what we really have talked about a lot at this conference, and in my lab we are fascinated with the goal of having accurate clocks that represent epigenetic aging, if not other aspects of aging. It's a bit of a complicated slide, but it does cover what we work on in my lab. This was from a review back in 2008 when we published a paper that showed that not just in yeast but in mammals, chromatin modifiers move around during aging, and that process can be accelerated by extreme cellular damage. And what we used in this case was the double-stranded DNA break, or DSB, and we used an enzyme called PPO1 to accelerate what looked like aging. And I think you've heard probably me talk about this before, so I'll go quickly. But we've used this system to understand the process of aging in mammals and how to reverse it. And just some aspects I want to point out. NAD is a cofactor, co-substrate, as you know, of sirtuins. And we found that upregulating SIRT1 gene expression as well as NAD levels in the cell can slow down this reorganization of the epigenome and prevent loss of gene expression patterns. And we did this in a mouse back in 2008 in neurons in the brain. But we've gone a long way since then, or come a long way. I want to report a little bit about MIB626, which is an NAD precursor. It's a polymorph of nicotinamide mononucleotide that is GMP pure. It's been in humans, and I'll update you on that and how that seems to work. And our goal there is to have a drug that will treat not just aging, but diseases of aging. And there are about, I think right now we've got 5 clinical trials up and running, and I'll share some results of that. So what about the clocks? We have a paper that's up online. It's not yet published. This is work by Patrick Griffin and Jian Li in my lab, 2 students who came up with the idea that you can greatly reduce the cost of sequencing by transposon tagging DNA samples, and we can pull 500 or more people or dogs or mice, and we've built some, and Patrick and Janet built some really accurate clocks for mouse tissues and in humans. You can see down below. Wherever I can, I give you directions to go follow up on this news. These are hot off the press human clocks from Patrick. They're really great. They've got all the attributes that you'd want. This is from human blood. We have brought in over 1,000 human samples from people, variety of ages, and you can see that the R values are really great, and the mid-AE of 3.5 years is not bad for a first start, and this is again just half the presses. The mouse ones look really good too. We also have phenotypic age clocks, which we've published on. If you're interested, you could check that out. How about slowing aging? Well, we've worked on activators of sirtuins for a long time. Resveratrol was the one back 20 years ago. We've developed some synthetic ones and some NAD precursors. MIB626 is, as I mentioned, an oral formulation of a polymorph of MIB626 that's stabilized crystalline, and it's gone through a lot of safety studies over the last few years, headed by this lab, Shally Bason's group at Brigham and Women's Hospital, and David Livingston runs the group at Metro Biotech, which is a Massachusetts-based company that's been making NAD precursors, both synthetic and natural, for the last decade. And full disclosure, this was spun out of my lab, and others, including Shinamai and Raj Apte. How do you measure what the effect might be? Well, one of the ways that Shally's lab did it was to look at endurance and strength within an MRI. And so this is an example of the machine that they built to insert the patients into the machine and measure their endurance, strength, and things like ATP, NAD, oxygenation. And some of the data that hasn't been published yet, but we're starting to talk about it, and this is their group's data, not mine, is that a number of things happen when you take 1,000 milligrams for roughly a month of this substance orally. And you can see here an example of some of the data. The repetitions to failure of that leg exercise significantly greater after taking MIB626. This is similar, or at least reminiscent, to the mouse data that we published in 2018 on endurance in mice due to increased vascularization via action on endothelial cells. Another nice bit of data, I would admit it was a surprise. A nice surprise is that Shally's group showed that lipids and cholesterol went in the right direction for improved health, as you can see here. HDL was not affected, which was also reassuring. What about acceleration of aging? Now, we do this for two reasons. One is to understand why we age, because if we can cause it, then we have some idea that we're on the right track. But we also want to know if we can have a model of aging that's much quicker, because typical longevity experiment takes way too long. And in organoids are way too young. When you build an organoid from a stem cell, an iPSC, they're age 0, which is not that useful. And so we've used this system, which you may know about. It's close to getting accepted, we hope. Last stages of this 12-year journey. Actually, I started this project with Philip Oberdorfer when I was 39, so I've aged a little. This project is the culmination of 20 labs around the world to see if we can displace chromatin modifiers in a way that mimics aging, and we've done that, and that's this work, which we call the ICE system. And we have cell-based systems and mouse systems to do that. And we'll use it in a minute, I'll show you later, but we can drive aging forwards as far as we can tell epigenetically, and the other hallmarks of aging accelerate as well, for the most part. The reversal, this is a very hot topic, as you all know. Typically we use a combination of 3 of the Yamanaka factors rather than 4. We leave out CMYC, and we're using other methods, chemicals now as well. We published this in 2020, so I'm not going to repeat that data, but we did use it to rejuvenate neurons in mice and restore vision in mice that had damaged or old retinas by targeting the neurons. And we find that we need all three of those factors, O, S, and K, shown down here in this vector that we can control with doxycycline. We have a TET on and TET off system if you would like to try it. This isn't injections in the eye, which was in the Nature paper. This one's the whole brain, and we're using a variety of AAVs now to infect different tissues and the whole body in efforts to not just accelerate aging, but take those mice and wild type and reverse them and see what happens. And this is work that I'm showing you from Xiao Tian primarily, and Jaeyun Yang. And their goal is to reverse the age of the brain and see what happens. And well, this was the paper I just wanted to remind you. And we had a lot of help from people in the field, some of who are at this conference. Morgan's here. Vadim was great help, Steve Horvath. There's a long list you can see here. I couldn't have done any of this without their labs as well. What was amazing about this was that we showed that there's a repository of youthful information that can reset a cell's epigenome. We measured gene expression on the right. You can see it's nicely reset proportionally to how the genes were expressed when they were young. This is RNA. And on the left, I think even just as impressive is the methylation patterns were largely restored as well. And the clock, which Morgan and Steve and Vadim helped us show, was retarded as well. And one of the interesting things in this paper is we found that damage to neurons accelerates epigenetic aging as well. So it's not just DNA cutting or time, it's extreme cell stress like a nerve crush that can do it as well. This has been commercialized or is being commercialized by Life Biosciences, a Boston-based company, also spun out of my lab. You can check it out, and you'll probably get the website if you spell the sciences correctly. There's an E in there. Life Biosciences was started in 2017. Bruce Cassander at Mass Eye and Ear has done a lot of the preclinical work and the safety work. We've now got a fair amount of data, and we're now in non-human primates with the goal of sometime in the next 12 to 24 months of starting work towards an IND and getting into humans if all goes well. And our goal is to reverse blindness in a variety of different disorders in humans. But it is remarkable if you reverse the age of neurons in the retina, you can restore vision and gene expression this way. Of course, I'm standing on the back of some very big names in this field who pointed the way. This is new data, unpublished. I was allowed to share this from Bruce Cassander's lab. He and I are infecting not the neurons here, but the retinal pigment epithelium cells, RPE, and these give rise to the photoreceptors. And these degrade over time, particularly in macular degeneration. And what Bruce has found is that he can actually restore the morphology and the function of these cells and restore eyesight back to a young state, similar to what happens when you rejuvenate neurons in the eye as well. The system in the lab now that we're doing is more human. We have inducible human neurons where we can differentiate them both into flat cultures and three-dimensional. And we do pro-aging with ice and reversal with OSK typically. Okay, I'll give you some examples of what we do. And this is work mainly by Xiao and Jay in the lab. We grow the nerves on these little culture dishes that can sense electrical signals. When we have a control induction of no OSK, there's not very good electrical firing. But when we rejuvenate them with the OSK treatment, just for a short amount of time, this just takes a little over a week, we see that there's a great induction of function, and it continues out the longer we go. This is day 50 that you're seeing on the right. So that's just neurons, that's human neurons. We've got Alzheimer's patient neurons as well. We can make these into little brain organoids. You can see here that we can turn on all of the 3 Yamanaka factors that we like in these organoids, which have a similar brain structure to ours. Great models for human aging. We can age these forward and get them to take on inflammatory and senescent signatures, which I think will be a useful model. In terms of reversal, we've done this in mice as well. We can now deliver the OSK system into various places in the brain, whole brain, or in this case on the right, the excitatory neurons. And in both cases, we get an improvement in learning in these old mice, which is what I think might happen. We might find out as a field is that diseases of old age, particularly in the brain, I'm thinking of like Alzheimer's, if you make the brain young, then the diseases just go away, because most of these diseases of aging are caused by aging. I mean, it's obvious to a lot of us, but to the rest of the world, it's not. So I want to thank the people who made this possible. There's a large group. The clinical trial group is at Brigham and Women's Hospital. There are 4 other clinical trials that are ongoing. There's one actually for Alzheimer's disease that's starting to recruit. There's one, well, I won't say the whole lot, but they're interesting ones. You can see that we've got collaborators both for exercise physiology, the hormones, and this is the MIB626 group at Metro Biotech. Here we have the people in my lab that helped or did major contributions. I want to point out the work of Jay, who did the ice mouse, and Matoshi. I want to thank everyone really, but I want to focus on, I mentioned Xiao, Jian for the OSK work, Yuancheng Lu, who left the lab after he published, rightly so. He's not listed, but he was the first author on that Nature paper. And I also want to point out Chris, who's doing reversal of senescence, Joao, who's working on a lot of the mouse projects. I could go on, I won't do that, but I do want to thank Patrick, who I probably didn't mention enough. He was the one that came up with this idea to do the time-seek clock. So many collaborators, it's impossible to thank everybody. These are the highlights, and I am so grateful for their advice, reagents, and friendship over the years. And the people who supported the work financially have been amazing too. So I'll stop there and take any questions. Thank you for having me.
Thank you so much, David. We have a question here in the front from one of our Inspire ambassadors. They're the high school students that are involved in the ARDD organization.
Hi, my name is Andrea. I was just wondering, so you said that you used 3 of the 4 Yamanaka factors when looking at eyesight rejuvenation. Have you tested any other combinations of these Yamanaka factors?
We tested all combinations, and we needed 3. 4 worked as well, but we were concerned about the loss of cellular identity, so we left c-Myc out. And those 3 are necessary. If we just do 1 or 2 combinations, it did not work. And if we gave them individually in different viruses, it still didn't work. So it seems like we need to package them into the same AAV, 3 of them, put them in a polycystron and get those in. Were there slight changes? Maybe, but the big difference was when we put in all 3. Now, we were trying for years different factors. So we tried nanog, and that didn't work very well. It was pretty toxic. And I mean, it sounds like we just tried something and it worked, but it was actually many years of Yuancheng's efforts to find this particular combination.
Great, thank you so much. It's quite late here, and we have a couple of questions more. Sorry, it's late for me, I guess. It's a couple of questions in the back there.
Hi, I was wondering if you also tested the data that you showed in RPE cells in pathological conditions, either genetic or chemical.
Yeah, so it's been done in a mouse model of macular degeneration with the iodide model, if you're familiar with it. Yeah. And it works well in that situation.
Okay, cool. Thanks.
There's another one from Lawrence.
Hi, David. I don't know if you can see me. It's Lawrence Ion from VitaDAO. I am wondering about the time-seek, if you can talk a bit about the implications there. How would it look like for the consumer? What does this enable? What are the downsides as well?
Yeah, I don't know of any true downsides. The cost is